Whether we like it or not, we share this planet with a number of microorganisms. We're going to discuss how they work with us as normal flora in causing infection and how we track down the kinds of infections they cause. Symbiosis is two organisms living together. We are outnumbered 10 to 1. There are 10 times more organisms living in us and on us than there are cells in our body. When the symbiotic relationship is mutualism, both members benefit. We benefit from our colon bacteria and they benefit from us. They get to eat our undigested food products and they make vitamin K for us. Commensalism is when one member benefits without significantly affecting the other. Staph epidermidis on our skin doesn't really do anything to us, and yet it can live off of our skin and our skin oils. It may give us a little bit of benefit because it's non-infective and it takes up space, so it may keep other things from growing there. The relationship we'll explore the most is parasitism. Here, one organism benefits and the other is harmed. The host is the organism that is harmed, and the parasite is the organism that benefits. When the parasite causes disease, we refer to it as a pathogen. We have normal microbiota or normal flora. There are certain axenic environments in our body. These are areas free of microorganisms. These are the sterile areas in our body. All of our closed body cavities, abdominal, thoracic, cranial, are all sterile areas. There are no microorganisms that are normally housed there. Also, below our larynx, the lower respiratory tract is considered sterile. It's not quite sterile, but it's considered sterile. Any of the surfaces and open body cavities are colonized with organisms, and these organisms are our normal flora. Resident flora is with us pretty much throughout life. It may be different from individual to individual, but it's ours and we keep it. Transient flora is with us temporarily. It may be with us a few hours, days, or months, and then disappear. You start out in the uterus sterile, but as soon as you hit the birth canal, you begin acquiring normal flora. With that first breath, you bring organisms into the upper respiratory tract. As you have skin-to-skin contact with people, you begin to pick up organisms on your skin. And as you eat and take in food and liquids, you begin to take microorganisms into your digestive tract. Opportunistic pathogens are normal flora that can cause infection. When they do this, they're referred to as an opportunist. There are basically three ways that normal flora becomes opportunistic. One is if normal flora is introduced to an unusual site. For example, E. coli is perfectly normal in our intestine, but if it gets into the urinary tract, it causes a urinary tract infection. Any normal flora entering a sterile body cavity would cause infection. If the person is immunosuppressed, then organisms can become opportunistic. People who are already sick with something, if they're malnourished, if they're under stress, this can suppress the immune system. Extremes of age, the very old and the very young have immune suppression, and anyone who's on chemotherapy or immunosuppressive drugs. And the third way organisms become opportunistic is changes in the normal flora. We have microbial antagonism or competition between our normal flora and anything else that tries to move in. If we remove the competitors, then other organisms can take over. The use of broad-spectrum antibiotics or long-term antibiotic use can destroy normal flora, allowing other flora to take over. Also, hormonal changes can change the balance of microbial flora. There have to be places in the community where infective agents can survive. These are called reservoirs of infection. There are basically three. One is animals. There are a few diseases that are called zoonoses. That's a plural. Zoonosis is a singular. These are diseases that infect animals and humans. There are about 150 zoonoses that we know of. Most animal diseases do not transfer to humans. There are just a handful that do. And typically this is transfer from animal to human. Seldom do humans give animals a zoonosis. Then we have human carriers. A carrier is a human that can transmit the disease to others. Some of these are chronic asymptomatic carriers. They've never had the disease, but they've somehow acquired it. Their immune system has kept it suppressed, but they can still give it to others. Also, people who are convalescing from a disease or are shortly after an infection may still be able to transmit the disease to others. They have no symptoms, but they're still capable of transmitting the disease. And then there are non-living reservoirs. These are things like soil and water, particularly if they've been contaminated with fecal material. In our exposure to microorganisms, we have contamination. Contamination is simply the presence of microbes in or on the body. We acquire them in food, drink, insect bites, sexual intercourse. Some of these organisms stay at the point of contact, they cause us no harm, and may become part of our resident normal flora. Some may stay at the point of contact, cause us no harm, only stay a short period of time, they're part of our transient flora. But some organisms introduced to the body overcome the host defenses, and when that happens, we have an infection. An infection may or may not adversely affect the body. In other words, you do not necessarily have to have a raging disease, but you will have a point of infection. Portals of entry are sites through which pathogens gain entry to the body. Normally, your skin and mucous membranes are intact and they're good barriers against defense. Mucous membranes additionally have mucus and some antibacterial chemicals on them. But if there are breaks in the skin or natural openings like hair follicles or sweat glands, organisms may penetrate the skin that way. Mucous membranes line cavities with natural openings to the outside. Any of these openings are how organisms get to the membrane. Your mouth, your nose, your anus, your urethra, any of those work as sites for organisms to get in. A few organisms can cross the placenta. The ones that most commonly cross the placenta can be remembered by storch: syphilis, toxoplasma, others like hepatitis B and maybe HIV, rubella, cytomegalovirus, and herpes. These are the things that usually pregnant women are screened for to make sure that they don't get or have during pregnancy. There are a handful of other organisms besides these, but these are the major ones. The parenteral route is any route that bypasses a natural opening. These would be punctures or injections. For organisms to cause a disease, they have to adhere to your tissues so they can grow to large enough numbers to make you sick, and then do some sort of tissue damage. Pathogenicity is the ability of an organism to cause a disease. Opportunistic pathogens are your normal flora that get in too large a number or get in the wrong location. But there are some true pathogens. These are organisms that strictly cause disease. They're not normally found in the body. Virulence refers to the degree of pathogenicity. This is the ability to infect the host and cause disease. Highly virulent organisms almost always cause disease. Less virulent organisms, like opportunists, cause disease only in a weakened host. Virulence does not have anything to do with the severity of the disease, just the increased ability to cause disease. Adhesion is one of the things an organism must be able to do if it's going to cause disease. Adhesion can occur because an organism has specialized structures, like suckers, hooks, or fimbriae. Tapeworms and protozoa have suckers and hooks. Some bacteria, you'll remember, have fimbriae. Ligands, also known as adhesins or attachment proteins, are lipoprotein or glycoprotein molecules that allow attachment to a cell surface receptor. This is how viruses and many bacteria attach to the cell they're going to infect. These adhesins may also determine host or cell specificity. Many organisms have both adhesins and specialized structures. Cells that have lost their adhesion factors are said to be avirulent. Without their adhesion factors, they cannot cause disease. They may lose these as a result of genetic change, or they may lose them as a result of exposure to disinfectants or antiseptics. Biofilms do not attach directly to a host cell, but rather to surfaces in the host. As a result of the changes that occur in the organisms, those streamers may leave the biofilm and cause damage in the host. Here you see an example of a microbe with an adhesin, or a ligand, and it is attaching to a receptor, a normal structure on a cell membrane surface. Virulence factors are things that damage the host or evade the immune system. Virulent organisms possess one or more virulence factors, and remember, virulence is the ability to cause disease. There are three major virulence factors: extracellular enzymes, toxins, and antiphagocytic factors. Extracellular enzymes allow for tissue damage and spread. Hyaluronidase and collagenase are enzymes that separate cells. Hyaluronic acid is sometimes called cell glue because it holds cells together. Hyaluronidase will dissolve this glue, allowing microorganisms to penetrate skin and mucous membranes. Coagulase allows the organism to coat itself in fibrin. This allows it to hide from the immune system. Now, while it's in this fibrin clot, it can multiply to larger numbers. Then kinases will digest blood clots. Some organisms produce coagulase first to hide, and then kinases so that they can break out. This allows for further invasion. Eukaryotic cells also secrete extracellular enzymes. Fungi secrete keratinase, which breaks down keratin in the skin, and some helminths secrete mucinase. Mucinase dissolves the intestinal lining. And here you see some examples of how these extracellular enzymes work. The bacteria secretes hyaluronidase so that they can get in between the cells and invade the deeper tissues. Here the organism secreted coagulase. We clotted off the bacteria. Then the organism secretes kinase. We have more bacteria than we had before, and this allows more bacteria to enter the body. Another virulence factor are toxins. Toxins are chemicals that harm tissues or trigger host immune responses that cause damage. When someone has toxemia, they have toxins circulating in the bloodstream. This means that the toxins may be carried from the initial site of infection to other parts of the body to do damage. Exotoxins are released by living organisms, and they're pretty specific. Exotoxins target a particular cell or group of cells. The good news is they're heat labile, so if we find them outside the body, we can destroy them by heating. Cytotoxins kill host cells in general or else impair their function. Neurotoxins specifically interfere with nerve cell function, and enterotoxins affect the lining of the gastrointestinal tract. All of these toxins have specific action, and we can give antitoxins to help counteract the toxins. Endotoxins are chemicals that are released at the death of the cell, during cell division, or during phagocytosis of the cell. Endotoxins have a more generalized activity. They cause fever and inflammation and sort of generalized symptoms. Because they are released at cell death, when you first start taking antibiotics, you may initially feel worse if you are infected with an organism that produces endotoxins because you'll have a massive toxin release. These toxins can get into the bloodstream, causing a toxemia. They will cause vascular dilation or vascular shock. And here you see some toxins. The exotoxin kills cells, allowing the bacteria to invade deeper. Endotoxins are released at phagocytosis. They're released by dead bacterial cells, and gram-negative cells are the ones that have endotoxins. Gram-positive cells tend to have exotoxins. The third virulence factor are the antiphagocytic factors. These inhibit the immune system response of phagocytosis and allow the organism to grow. Capsules are an antiphagocytic factor. You'll remember that capsules are a thick glycoprotein layer. The glycoproteins do not particularly stimulate the immune response, plus they cover the attachment proteins for phagocyte attachment. Antiphagocytic chemicals are released when the organism is actually eaten by a phagocyte. These chemicals inhibit the phagocyte from destroying the microorganism. These organisms may actually live and divide inside the cell. Ultimately, the cell dies, and then a great many organisms can be released. Some of these antiphagocytic chemicals actually release chemicals that kill the cell. These are things like leukocidins. So here you see phagocytosis blocked by a capsule. The phagocytic cell just can't wrap around it, just can't take it in. And here you see one that was taken in, but the lysosome can't bind here, can't add the digestive enzymes and kill the cell, and the cell reproduces inside the phagosome. When this cell dies or is potentially killed by another antiphagocytic chemical that's released by this organism, instead of one organism, we will be releasing multiple organisms. Diseases manifest by showing signs and symptoms. Symptoms are subjective. These are things that cannot be confirmed. Pain, a headache, nausea. There's no way to confirm that these are actual. Signs are objective. They can be observed or measured. Temperature, for example, blood pressure, these can all be measured. Signs, symptoms, and lab tests can usually lead to a diagnosis of the infectious disease. A syndrome is a group of symptoms and signs that, when we see them all together, characterize a particular disease or abnormal condition. AIDS, acquired immunodeficiency syndrome, is that cluster of signs and symptoms that include a positive HIV test, a reduced CD4 count, and the presence of certain marker diseases, certain pneumonias, cancers, or gastrointestinal diseases. Asymptomatic or subclinical diseases are diseases that go unnoticed because they have no obvious signs or symptoms. Sometimes you can do tests and you indicate that there's a problem, but you may not be able to pinpoint what the problem is. The cause of disease is known as etiology. Not all diseases are infectious. We have congenital diseases that people are born with, degenerative diseases, nutritional, endocrine, mental, immunologic, neoplastic diseases. None of these are caused by microorganisms. The etiology of infectious diseases relies on Koch's postulates to demonstrate what the infectious agent is for the disease. You'll remember that Koch's postulates, you had to find the organism in every case of a disease. You had to isolate the organism from a sick animal and grow it in pure culture. You then use the cultured agent to cause disease in a healthy organism and then recovered the organism from the second sick host. There are sometimes exceptions to Koch's postulates. Sometimes you're unable to culture the microorganism in the laboratory. This is the case with Mycobacterium leprae, the causative agent for leprosy. Sometimes there are multiple causes of a disease. Dental caries, for example, is caused by a group of organisms, a biofilm of organisms. And there's a form of liver cancer that seems to be stimulated by viral infection, but you have to get two viruses. You have to have both hepatitis B and hepatitis D. Either alone does not seem to stimulate liver cancer. If humans are the only host, we have ethical concerns about finding that second host to give it to. Sometimes we have a disease like pneumonia, and there's more than one causative agent. We have bacterial pneumonia with several different bacteria causing that, and we have viral pneumonia. And occasionally, a pathogen is just not looked for. It's ignored. Helicobacter pylori causes 90% of the gastric ulcers in this country. And yet everyone assumed that gastric ulcers were caused by stress, spicy food, certain medications. It wasn't until a doctor looked for an organism that one was found. If we can't use Koch's postulates, we can sometimes use epidemiological studies. These can give statistical support to show the cause of a disease. Dr. Snow was able to show that a particular well in London seemed to be the source of the cholera outbreak in a certain part of London. He didn't know the causative agent was Vibrio cholera, but he knew that that well seemed to be where people were getting sick. When we look at the stages of an infectious disease, we start with the incubation period. This is the time between getting the infection and the first symptoms or signs of the disease. The length will vary considerably. It will depend upon the virulence of the organism, how much of it you initially got, the infective dose. If you were given a whopping dose of organism, it won't take long for it to get to numbers to make you sick. If you just got a little bit of it, it's going to take a while for it to incubate in there. What's the condition of the patient? Are they relatively healthy or are they somewhat run down? What's the nature of the pathogen? Does it have a number of virulence factors or not? How long does it take for the organism to reproduce? And what's the site of infection? Some sites are more resistant to infection than others. Some organisms have a typical time. Most of the childhood viral diseases had 21 days. During the incubation period, the patient may be infective for others. This may be a carrier period. The prodromal period is pretty short and may not exist at all. This is that short time when you think, I'm coming down with something. I think I'm coming down with something. You start to feel a little bit not right. The illness is when all of the signs and symptoms of the disease are evident. Your immune system is starting to respond but hasn't come into full play yet. During the decline phase, the body returns to normal. Here, your immune system has mastered the disease and you're getting over it. And then you go into the convalescence period, which is the period of recovery. This also varies in length. Sometimes it's quite long, sometimes it's relatively short. Again, this is when someone can be a carrier. They really aren't showing signs and symptoms of the disease, but they may be infectious. There is one more consequence or possible stage of an infectious disease, and that is death. Not everyone recovers from every infectious disease. We talked about how do they get in, how do they get out, what's the portal of exit. They pretty much get out the same way they got in. Any secretion, excretion, blood, body waste, wound drainage, exudates, any of those things carry infectious diseases from any normal body opening or any wound in the body. How do we transmit diseases? Well, we have contact transmission. This can be direct contact, that's person to person or mucous membrane to mucous membrane. This is between hosts. This is direct contact between two hosts. Direct contact is also considered for any diseases that cross the placenta. And then there may be autoinfection. You may have an infection in one part of your body, and then as you touch that and touch your eyes or touch your nose or put your fingers in your mouth, you take it to a non-infected site, or you keep reinfecting yourself. Indirect contact is through fomites. Fomites are inanimate objects that are shared by hosts, so you don't have to have direct contact with the person, just contact with something that they've handled. This is why you should never drink out of the same glass as someone who's sick. That becomes a fomite. And droplet transmission is when droplets, usually from the respiratory system, are picked up within 1 meter of the host. Droplets are usually pretty good sized chunks of stuff. They don't travel very far, and they may settle down on surfaces or on dust particles. Vehicle transmission is when something like air, water, food, body fluids handled outside of the body become the way that the infection is transmitted. Airborne transmission occurs when aerosols are produced. Now, aerosols are very fine droplets that can stay suspended in the air for a fairly long period of time. They too may also settle and become pathogens on dust. Aerosols can be created by coughing, sneezing, air conditioning, sweeping the floor. Anything that stirs up dust can put aerosols back in the air. Waterborne transmission is usually through the oral fecal infection route. In other words, water has become infected with feces and we're drinking the water or using the water somehow. Foodborne is when pathogens are in or on the food. Inadequate processing, undercooked food, poor refrigeration, all may allow organisms to grow to large enough numbers to make us sick. And then body fluid transmission. When you're handling body fluids outside of the human body, you should handle them all as if they are infectious, because they very well may be. And then finally, we have vectors as a form of transmission. Vectors are animals that transmit disease from host to host. Some vectors are biological vectors. They not only transmit the pathogen, but they're also part of the infectious agent's life cycle. For example, malaria is transmitted by the mosquito. However, the malaria organism completes part of its life cycle inside the mosquito. Mechanical vectors are not required in the life cycle of the organism. They simply pick it up and carry it around passively. Things with sticky feet like flies and roaches are excellent mechanical vectors. Diseases can be classified a number of ways. One of the ways is with the rapidity of onset and the length of duration. An acute disease has a rapid onset and lasts for a short period of time. Frequently, the symptoms are fairly severe. A chronic disease has a slower onset, lasts a longer time, and has less severe symptoms generally. And a subacute infection is somewhere in between. Doesn't come on as fast as acute, comes on a little faster than chronic, Lasts a little longer than acute, but not as long as chronic. There's a disease called subacute bacterial endocarditis. This is when bacteria live on the heart valves and are shed intermittently as the heart beats. This causes mild fevers, so they're not very severe symptoms. It may go some time before it's diagnosed. A latent disease is one in which a pathogen remains inactive for long periods of time. Herpes is a good example of this. A communicable disease is one that's transmitted host to host through either direct or indirect contact. A contagious disease is one that is very easily transmitted. Host to host contact is not necessary. And there are certain non-communicable diseases. These are when there are organisms outside the host that we acquire from the environment, that once we've acquired them from the environment, we can't pass them on to anyone else, or diseases that are from our own normal flora. We can't give those to anyone else. Epidemiology is the study of disease in populations. It's involved in identifying the source of disease. In epidemiology, they're concerned with the frequency of a disease and the occurrence of a disease. When we talk about frequency of a disease, we talk about incidence, which is the number of new cases in a given time, and prevalence, which is the total number of cases in that same time period, both new and existing. By keeping up with things like incidence and prevalence, you can track things like how long it takes to get the disease, how long the disease lasts, what's the recovery period, things like that. The occurrence of disease has to do with how many occur at any given time. Some diseases are endemic to a region. That is, there's always a certain number of cases in that region at any given time. Histoplasmosis is said to be endemic to the Ohio River Valley. This means that at any given time there are a handful of cases somewhere in the Ohio River Valley. A sporadic disease is one that occurs in random outbreaks. Every once in a while we have an outbreak of meningitis. There's a half a dozen cases and then it goes away. We may go several months, several years, and then there's another little outbreak of meningitis. 5 or 6 cases, that goes away. So there are long periods, there's no pattern to when it occurs. Epidemic is when we have more than the expected number of outbreaks of disease. Now, usually people think epidemics are thousands and thousands and thousands of cases, but that's not the case. An epidemic of botulism is one person because we expect no botulism. Sometimes we can have a normal number of diseases, a normal number of cases, but it's at the wrong time of year. For example, we expect a certain amount of influenza in December, January, February. We do not expect to see influenza in August, but if we start seeing it in August, that could be an epidemic. And a pandemic is a worldwide epidemic. AIDS is pandemic. There are different kinds of epidemiological studies that can be performed. One type is descriptive epidemiology. The researcher tabulates all of the data concerning a disease. Mostly they're trying to identify the index case, or what they sometimes call patient zero. Sometimes they can't do that because that individual may have gotten well and we can't track the disease back to them, but that's what they're trying to do, trying to find out everything they can about that disease. In analytical epidemiology, they investigate the disease in detail. They use the descriptive data, and they try to determine the probable cause, the mode of transmission, and a possible means of prevention. This is used when Koch's postulates can't be used to try to identify the source of a disease. In experimental epidemiology, they're testing the hypothesis concerning the cause of disease. This is when they will use Koch's postulates if they can. Another way that they can use experimental epidemiology is by trying to treat a disease they think is occurring. Chlamydophila pneumonia was believed to be a cause for atherosclerosis. To test that hypothesis, they took a large number of people and they treated them with an antibiotic that should have killed the organism. Even though they treated all these people, they all still had atherosclerosis. So it appears that this particular organism does not cause atherosclerosis. Nosocomial diseases, or as your book likes to call them, healthcare-associated infections, are diseases that are acquired when people are in healthcare institutions. Exogenous diseases are those that are acquired from the healthcare environment, equipment, air, anything they come in contact. But there are a number of endogenous nosocomial diseases. This is when the normal flora of the hospitalized patient is actually the cause of the disease. Iatrogenic infections means that they're doctor-induced. That's what iatrogenic means. But what these infections are is they result from treatment. We use indwelling catheters. We do surgery. We do all kinds of procedures we never used to be able to do. The more invasive the procedure, the more likely there is to be an infection as a result of it. Superinfections are what we see when there is antimicrobial use that inhibits the normal flora. There are several factors that influence nosocomial infections. One is the fact that there are numerous pathogens in healthcare settings, so the patient is exposed to a lot of organisms. There are biofilms, there are antimicrobial resistant strains, all kinds of nasty stuff in healthcare institutions. We also have the perfect population to get sick. People who are in hospitals are not healthy. They have weakened immune systems and are very susceptible to disease. And then we have a lot of people. We have transmission from patient to patient. We have staff to patient transmission. We have visitor to patient transmission. At one time, children under the age of 12 were not allowed into hospitals to visit. The reason was not because they weren't old enough to see something, but because kids under 12 are carrying a lot of microorganisms. They're big reservoirs for microorganisms. In controlling nosocomial infections, there is absolutely nothing in the world better than good old-fashioned hand washing. This is the one thing that they push, push, push. You cannot go into a healthcare facility and not see some sign up about hand washing. And this is actual honest-to-God soap and water hand washing. Hand sanitizer works from time to time, but periodically you need to wash your hands. Also, we can use disinfectants appropriately. Make sure we're using medical asepsis at all times. General good housekeeping helps. And using isolation techniques to keep healthier people away from sicker people. Most healthcare facilities have an infection control committee. This is a group of people who meet from time to time and look at infection rates in different parts of the facility to see if they can see any trends. Epidemiology and public health go hand in hand. The local, state, national, and world agencies all share data. All of these agencies are collecting data and they share it with each other. We start with the city and county health departments. There are state laws that govern what diseases must be reported by law. The state agencies report to the Centers for Disease Control, and the CDC is one branch of the United States Public Health Service. The United States Public Health Service and the CDC works with the World Health Organization, and this is the organization that coordinates improvement in world health. The role of public health agencies includes enforcing standards of cleanliness in food and water supplies, reducing disease vectors in reservoirs. For example, it's the public health agencies that try to kill mosquitoes every year because mosquitoes are a big vector. They help establish and enforce vaccination schedules. They also locate and prophylactically treat individuals who have been exposed to disease. And they establish isolation and quarantine measures to control disease spread. It is the public health department that makes sure water is fit to drink, that it's potable water. And you must use potable water for drinking water, for hand washing water, for anything like that. Also, it's the public health department that inspects food processing places, restaurants, food supplies to make sure that they are safe. And they're involved in public health education. Every year you see a number of public service announcements from the public health department about hand washing around cold and flu season, getting vaccinations, all kinds of things that would help the population be healthier.